Hey, good morning. Woo, that's a pretty live mic. I think I could almost do it without a mic in this room, but I think we're recording, so I have to use the mic. Yeah, okay. Okay, everyone's here for financial planning and investing in your future for retirement. Okay, good. We're going to talk about a couple of emerging contaminants today. I'm going to chat with you about PFAS, and my colleague and friend Brian Thompson is going to chat with us about 6-PPD-quinone. We're going to have a word also from, in the middle of the presentation, from Alex Brewer, our laboratory director, to let us know a little bit about what's coming online at our analytical lab in Corvallis. So I said I wasn't going to run to this table, but I already did. Okay, so we'll get started. This presentation will be available online, and you'll see a lot of links and these kinds of things that you can click on. So if you want to download the presentation and go into more detail on things, feel free. We've got several publications out here. We've got a strategic plan, strategic roadmap. We've got updates and a recently released document on looking at options for disposal of PFAS. Spoiler alert, there's no silver bullet for disposal or destruction. I'll get to that in a little bit. Okay, unless you've been really not paying attention, you're probably at least heard of PFAS. And the long name for it is per and polyfluoroalkyl alkyl substances. You can see there at the top. And this is a group of man-made chemicals that I think they were first started to be produced around at the end of World War II, when all of the halogenated compounds, various chlorinated solvents, et cetera, started to be created. And they were found to be really useful. One of the big uses we had was aqueous film forming foam, which is a firefighting foam. And we've used a lot of that, particularly at airports and military bases. And I was thinking, oh my gosh, why'd you use this terrible stuff? And the military described to me, I was talking to one of the Air Force folks, and they said, look, we were watching pilots die on the tarmac. We couldn't get to them. This firefighting foam was a lifesaver. It was incredible stuff. It changed everything. So, you know, at the time it really helped people, but now we're finding it's quite toxic and it's a problem. So it's been used in
industry. It's just been used all over the place, and we're finding different sources of it: incineration and thermal treatment facilities that have been used to either regenerate granular activated carbon or try to destroy PFAS. We're seeing it leaching from landfills, wastewater effluent, land application of biosolids. This may be an issue for tribes. We'll mention that a little bit later. We have PFAS everywhere. It's been used in consumer products. Think Teflon, right? How many people here own a Teflon pan? More than one, right? Throughout your life. So cookware, food packaging, you know, those little white food boxes that you take home, you notice how they're kind of shiny on the inside? Yeah, that's the waterproofing. It's plastic and PFAS. Stain and water repellent fabrics, right? It's all over the place. And so the bad news about PFAS is if you're going to spill a chemical, it has all the aspects that you don't want in a chemical that's going to be released in the environment. It's widespread. We're seeing it all throughout the ecosystem and throughout our lives, all these things I listed. It's toxic, highly toxic. We're regulating it at part per trillion levels. I mean, we've all heard of parts per billion, but parts per trillion. And when we did initial assessment, we were looking at part per quadrillion toxicity. Yeah, I mean, that wasn't even in our, in our vocabulary. It's bioaccumulative, meaning it can build up in your body over time, stay in your body long term. It's mobile in the environment, so it can move through the air, soil, and water.
and it does not break down quickly, right? All the things you don't want in a chemical. So bad stuff. EPA is trying to do something about this, and we have a three-prong approach. And this is basically where I'll cover things pretty much. We're doing research. We're trying to restrict the use of PFAS, right? Get in front of it. If you can cut off the sources, then you're ahead of the game. And once the horses are out of the barn, we're going to clean it up, right? Remediate. So that's the way EPA is approaching this. Research, restrict, and remediate. I'm not going to go into a lot of detail on the research, even though I'm the Regional Science Liaison. This is kind of my gig here. But these are the kinds of things that we're doing. And what you can see is we're doing research on all aspects of PFAS. Detection methods, toxicity, exposure, how to clean it up. We're looking at innovative treatment technologies. We're using the Small Business Innovative Research Program to help us better understand ways to analyze PFAS in real time. We're also teaming up with Department of Defense and Department of Energy, and we have the collaborative research program, because they have a lot of money that they're pumping into research too, because let's face it, Department of Defense owns a big part of this problem, right? They've used a lot of PFAS. So that's all I'm going to say about research. Let's talk about restricting PFAS use and sources. This is important. We need to restrict the sources, get ahead of this, right? If you're not using it, it doesn't enter the environment, we don't get exposed. CDC runs the NHANES program, and they do sampling across the nation. And they've been looking at blood levels of the most common PFAS in people. This only goes up to 2018. They only do this sampling once in a while. But you can see for the various compounds, PFAS, PFOA, PFHXS, and PFNA, this is micrograms per liter, parts per billion. These levels are above what we're regulating in groundwater. And this is the average concentrations in our bloodstream around the country. That's kind of scary, isn't it? With Halloween coming up, right? These concentrations are higher than you should drink in water, so vampires beware. All right, so here's some of the things we're doing to cut off the flow of PFAS into our world and into our lives. We've added it to the TSCA, Toxic Substances Control Act. We have SNRS, which kind of sounds like a Dr. Seuss regulation to me, but it's significant new use rules, so we're limiting new uses of PFAS. We've got the toxic release inventory. We've added 172 new PFAS, requiring industry to track any releases. That's the toxic release inventory, so so we can understand who's spilling this stuff, where it's being released. We've removed PFAS as an inert ingredient in pesticides. It was used to line the pesticide containers and keep them from leaking. We've got a national PFAS testing strategy requiring manufacturers to provide PFAS data, effluent and pre-treatment standards. You get the picture. We're trying to really get ahead of the game. Risk assessment in biosolids, that's going to be coming out at the end of the year. Office of Water is working on that. They promised it by December, we'll see. PFAS in oil, and I'll talk about this a little bit more in detail, and I think this is going to be important to tribes. We recently released updated fish and shellfish consumption advisory numbers for PFAS. All right, so here we are, restricting PFAS. Here's the big news, new for 2024. We have MCLGs. This is the goal. This is what we would like to see. We think, and this is the maximum contaminant level, the actual national primary drinking water regulation. These numbers here, this is what we think drinking water treatment plants can actually accomplish. And this is what we're going to hold people to, including tribal public water supply systems. You have 5 years to come into compliance, but you're going to have to meet these numbers in terms of treating for PFAS in your drinking water systems. And our drinking water folks, I want to identify our tribal drinking water folks. There's Carly back there. If you could go ahead and raise your hands, EPA tribal drinking water folks, yep. Ian, Anna, there we go, yep. So those folks can really help you with what's happening and what you're gonna have to do. And we have money for sampling, we have money for infrastructure. I'll mention that again in a couple slides, but there is money to help you out. All right, we're building on our National Primary Drinking Water Standard, the MCL, restricting PFAS in drinking water. We've got EPA 537.1 and 533 test methods for 29 PFAS. We've got a drinking water treatability database with options for removing 37 PFAS compounds. This is really helpful, particularly if you're trying to treat private wells and things like that. We've got a drinking water tribal set-aside program with a fair amount of money available to help you build up your capabilities for drinking water treatment. And conveniently, right after this session, they're going to tell you all about that program. So if you're running a drinking water system and you're worried about PFAS in your drinking water system, stay in this room, 10:30 to 11:20. They're going to tell you about this. Very important. Applications are due by December 6th, right? So you want to know about this and you want to get on it if you're going to get part of this money. And coming soon, we're going to have a PFAS treatment fact sheet from Region 9. I want to hand the mic over to Alex for a moment. Alex Brewer, our lab director.
Thank you. Yeah, I guess I don't really know what to say. I'm the Region 9 lab manager out of Corvallis. I know you're thinking Corvallis, Oregon. I know you're thinking, well, that's not Region 9, but we're the Region 9 lab. It's not a problem that we're in Oregon. We ship samples all over the place. So I wanted to talk a little bit about what we do, and 533, maybe 537, 1633 are PFAS methods that we have in progress and the ones that we have currently online, or freshly online, I should say. Yeah, so generally speaking, we're an analytical lab. We're just like any environmental commercial lab. Some people may say production lab out there, but we're EPA. So we work with our EPA clients that work with tribes to take samples in to run them, to get people their data. So that's what we do. That's what we're good at. We're good at quantitative analysis, getting people their data in the highest or with the highest quality possible. Okay, so you may currently have samples coming through the lab, even for PFAS, whether it's subcontract, contract, or actually directly through the lab. We work with a lot of tribes currently, and we also work with a lot of EPA clients. So if you have questions about the lab, just generally speaking, you could just shout them out or just talk to me afterwards. Our PFAS methods, 533, drinking water, 537, drinking water, they're slightly different. There is some overlap, but they're pretty close in terms of what they are and how they're completed. 1633 is for dang near anything else. So a lot of soils, tissues, leachate, which is stuff that comes out of landfills. They're all LC triple quad methods. So what that means for the person that's submitting samples is that it's going to be expensive. Because it's part per trillion, it's somewhat difficult, but part per trillion, part per billion, not that difficult for an analytical chemist or a commercial lab. So I just wanted to talk about how we're bringing up 533 and we're ready for accreditation. So for our EPA partners, we're really close to having them come through us and being done that way, which should save money in the long run. Are there any questions? Oh, go ahead.
What's the detection limit?
Detection limit for which method?
Give me a range.
Well, it's obviously going to be lower than 4 for PFAS, if you want to go back.
Lower than 4
Nanogram per liter.
trillion. Okay.
So, yeah, I believe it's like, off the top of my head, I don't really remember.
But it has to be below this.
It's below 4.
Okay.
It's going to be
Yeah. Yeah. And like I said, for nanogram per liter, that's not terribly difficult for us, but the tricky part for any of these methods is contamination. So that's what will sort of raise up the cost, that and what equipment we use. So it's a SPE method, which means solid phase extraction. There's a lot of sample prep, so a lot of time goes into that. And it's a LC triple quad method, which means it's going to be expensive. So all those things put together mean fairly expensive to run, just in comparison to most other methods. So for like metals, for instance, metals are a lot cheaper.
One more question, and then we'll save questions for after. Yeah, go ahead.
I'm doing a surface water program at drinking water. Could you use the same testing methods for surface water?
Surface water, I believe, is 1633. I mean, you could, yeah. People would really prefer you to separate out drinking water and surface water or any other water, for instrument sake and sample prep sake. So yeah.
Great, we'll take some more questions for Alex. Think about your questions, and at the end of this, we'll take a few more questions, and at the end of the whole session, we'll take more questions, even more questions. The, you know, part per trillion, that's like a few grains of salt in a swimming pool. I mean, it's like magic that, you know, Alex and his folks can even find that. I mean, you dip your finger in, does this taste more salty? No, it does not. So, you know, it's a tiny, tiny, tiny amount. And when he talks about cross-contamination, I was saying, where's PFAS, right? Waterproofing, it's all over the place. So if you're taking a sample, and you get some household dust in it, is there PFAS in household dust? Yes, there is.
The thing on that slide to be concerned about is the zero. For any analytical chemist, if you say zero, that's, zero.
That's part per Googleplex. Yeah. So, I mean, this is what we would like it to be based on health calculations. This is what we think can actually be achieved with modern day treatment, water treatment technology. And Emmanuel just walked in. I just, not to put you on the spot, but she runs our infrastructure programs. She's the one who can help you get money for treatment technology. And Carly's the one who can help you get treatment and also. There you go. I'm gonna put them on the spot. I've got their names on the slides. What am I doing here? Where did I go wrong? Okay, there we go. All right, so as I mentioned, I'm going to put them on the spot. You can see Joy's name here and her phone number and email. So contact these folks. They can help you with technical assistance. They can help you get sampling. They can help you get money for drinking water treatment. And we're supplying, even though Alex doesn't have the method up and running yet, we're supplying sampling through a subcontractor. And once Alex gets it fully up and running, we're going to run it through our own lab. So we're here to help you with PFAS. You're not out there on your own. So let's talk a little bit about the data collection. We've got tools out there that you can look at. We've got the, we're actually out sampling wells across the country under the unregulated contaminant monitoring rule. It's so hard for me to say. Unregulated, I always want to say uncontaminated. Unregulated contaminant monitoring rule, UCMR, and we do this every couple of years. And we're out sampling for 29 PFAS right now and lithium. You can look, you can download this and hit the links and look for more info. We've got PFAS analytical tools online, plotting data across the country. We've also got tribal PFAS monitoring results. You can see there's no Region 9 monitoring results on here because a number of tribes have asked us not to use or not to publish the monitoring results. Okay, so that's important, and we will not publish them, at least on this database. But if you're going to use those results, and we recommend that you do, if you're going to use those results for your initial round of compliance sampling, which is something you have to do to comply with the MCL, then we do eventually have to report those as compliance samples. So it's not that you can have your data completely ignored. You're running a public water supply system, your data is public, but we're gonna put it in the appropriate place, not necessarily listing it on this database. And that's, you know, important. People need to know what's happening with their drinking water, right? So it's important for it to be publicly available at some point. We've also, as I mentioned, got this new fish and shellfish monitoring recommendations. And we're recommending monitoring two categories. Monitoring for advisory. These are chemicals that we already know something about the toxicity. And so you should be monitoring these. And if you see them above toxic levels, you should be basically producing consumption advisories. These are ones that we should think you should be watching for. We think they're a problem, but we don't have toxicity on them yet. But if you see them, you probably want to think about advisories as well. So, as I said, we're recommending contaminants to monitor and analyze for fish and shellfish advisories. When these contaminants are in high enough concentrations, you can issue consumption advisories for water bodies. This is important. Tribes can create their own fish and shellfish consumption advisory programs. And I think this is helpful, particularly if you're doing subsistence fishing and you might have PFAS in the fish, right? And do you think your tribal members might be eating more fish than the average person? Then, you know, you might want to look at that and create your own consumption advisory levels appropriate with consumption levels for the population that you're looking at. We've even seen in some of the Great Lakes, and this is kind of scary, a couple areas the PFAS is so high I was talking to folks in our Duluth laboratory and they're monitoring the Great Lakes. And they said in a couple areas the PFAS levels in the fish is so high because it's bioaccumulative and the fish are at the top of the food chain. So all the other little critters are picking it up as they eat it. It's building up in the fish's body. They said it's so high you probably should only eat one fish a year. One fish a year. Okay, remember? A few grains in a swimming pool, it's building up in these fish. So this is serious business if you're eating a lot of fish. And so we've got guidance online to help you figure out what to do. All right, so research, restrict, remediate. We're into the remediation portion. This is new hazardous substance designation. And so it's requiring all of these things. And probably most importantly, compels potentially responsible parties to clean up. And we can do EPA response without proving imminent and substantial endangerment. So we can move much more quickly on PFAS releases. We're also trying to do the same thing under RCRA authority. So this is Superfund authorities. We're looking at RCRA authorities as well to move forward on requiring cleanup. This is also going to help us with transportation, storage, and disposal. Thank you. I wanted to show for our enforcement discretion, I want to be clear that we are focusing on PFAS for enforcement, but we're focusing on holding responsible those who significantly contributed to the release of PFAS in the environment. We're talking manufacturers, right, DOD, those kind of folks. We don't intend to pursue entities where equitable factors do not support actions or costs under CERCLA. Farmers, water utilities, airports, local fire departments. Some of you have local fire departments that may have used PFAS as well. You're not on our list for enforcement at this time. More on remediation. We've got revised regional screening levels. This is all online. If you have a Superfund site, work with the Superfund project managers. They can help you through this. And we've got aquatic life criteria for those of you who are concerned about your rivers and streams. So this is, and these are now final, the aquatic life criteria. These can be used, the MCLs can now be used as ARARs to drive Superfund cleanups. So we're covering Superfund as well. I did say I would mention destruction and disposal. Again, there's no silver bullet
This is the table of contents from the document. Basically, we're saying there's 3 main methods. Underground injection, this is probably the best method if it's a liquid waste for disposal at this point, but it's not available everywhere. So this is deep well injection, and it's certainly not available everywhere. The conditions are not always available. Permitted hazardous waste landfills, the leachate controls are important, and permitted hazardous waste combustors. This is important because people are sending PFAS contaminated materials to combustion facilities that may or may not have enough residence time or high enough temperatures. It's a big problem. So you need to use the right kind of facility. Okay, PFAS destruction and disposal. Not going to go into too much detail here, but we're working to do a lot of research to better understand PFAS destruction. And we really want to understand incineration. And this is our test incinerator at the Research Triangle Park facility in North Carolina. We're also looking at PFAS in air. I don't think people are thinking about that too much, but it is in air. So we're trying to do additional research to better understand disposal, destruction, and all the other problems. I'm Matt Small. I'm the Regional Science Liaison. You're welcome to reach out to me. There's also a couple of websites here that are really helpful. We can take 2 questions and then we'll move on. Yes. I can share.
Thanks. Regarding PFAS in fish tissue, are y'all doing research on where PFAS accumulates in different tissues? That's my first question, and then I have another one.
Oh, you're going to take both questions? Okay. The funding question, I don't do the funding. I would say, can you stick around for the next session? Oh, for tissue monitoring. That might be an Alex question. So, I'm sorry, what was your first question?
Are you conducting research on where PFAS?
Oh, yeah. Okay. So at this point, they're just doing kind of the basomatic approach, right? Stick a fish in a blender and look at the total PFAS concentration. But we're moving now to look at separate PFAS tissues. We're seeing it depends on the PFAS itself. Some are preferentially in the skin and the organs. Some may be more in the muscle meat, but in general, the fillets are probably safer to eat. But you should probably still be sampling for that list of compounds that I showed for the fish and shellfish advisory. And then your other question is about funding for 1633 fish tissue sampling and analysis. Yeah.
Sorry. As we know, folks are all resource limited, and the recommendations for monitoring are great. But will there be resources for tribes to support monitoring for fish tissues? Because if you've done that monitoring, like the water samples are expensive, tissue's much more gnarly and worse and more expensive. And then will EPA be providing support to tribes for advisory development?
Well, we can certainly advise you. I don't know if we're going to be covering the cost of 1633 at this point or not. That's probably a Carly question, and she had to leave. I don't know, Emmanuel, is there thoughts about covering fish tissue analysis?
Yeah, I think I'm brainstorming here, but we have a clean water set aside to address emerging contaminants. So if there are concerns around PFAS in your surface waters and fish tissue sampling is the suggested kind of monitoring approach, I think that could possibly be eligible for that program. We're going to talk a little bit more in the next session on all the funding resources around both addressing drinking water, emerging contaminants in drinking water, and emerging contaminants in clean water.
Is your question quick?
Yes. Are you measuring PFAS in?
Are you measuring PFAS in other foods like beef, lamb, vegetables, or can people not eat anything?
That's an excellent question, and it's something that tribes should think about. And this is why, because some tribes are receiving biosolids from nearby municipalities, and those may be high in PFAS. And if you're land spreading those, particularly for any kind of agricultural use or any kind of grazing, the animals can pick up, and the food itself, the plants grown on those biosolids can potentially also pick up the PFAS, and it can bioaccumulate in the plants
in the animals and in us? So that's an excellent question. I don't know if we're going to be providing that. I think, like Emmanuel said, stick around for the next session. I don't know if that would fit under Clean Water Act because it's more of a biosolids kind of thing. Maybe it might. We'd have to think about that. So, yeah, I don't know the answer to that one, but it's a good question and it's something tribes should think about. Are you ready, Brian? Yes.
Matt, before Brian gets started, on the funding, is that just for regional lab or is that ORD funding as well? So if you were to go to ORD and get some funding there.
That is not ORD funding.
Or is it just the regional?
Yeah.
This is regional funding. It's a set aside off the Clean Water Indian set aside to look at emerging contaminants that in your surface waters. It's all leading to, this is project specific, so it wouldn't be ongoing monitoring. It could support a specific project that would lead to infrastructure mitigation. So I want to be clear. It's not like the 106 may be more appropriate for ongoing monitoring. But if this is a project specific monitoring to characterize PFAS in a surface water that would lead to mitigation down the line. That's the idea behind the program.
And if you do have a research project, if a tribal member or tribal contact has a research project, you can contact me and we can talk about resources and options for working with our Office of Research and Development.
Okay, let's go ahead and jump to 6-PPD-quinone. And I'm Brian Thompson. I work at US EPA in Region 9. I work in the water division and in the watersheds section. The watershed that I mainly work in is the Central Valley of California and the San Francisco Bay Delta watershed. And in that capacity, I do a lot of work on water quality issues related to flow and temperature and salinity, which all impact salmon. And so it was kind of a natural jump for me to get engaged in 6-PPD-quinone because of its link to salmon, especially coho salmon mortality. Okay, so so just a brief introduction on 6-PPD-quinone and kind of the issues it's been causing. So 6-PPD, the kind of the parent chemical of 6-PPD-quinone is an ingredient in the manufacturing of tires that reduces the extent to which the rubber in tires breaks down due to reacting with ozone. 6-PPD-quinone is one of the byproducts formed by this reaction with ozone and is subsequently released attached to particles, tire wear particles into the environment. And 6-PPD has been in use since the 1960s. So even though this is a chemical of emerging concern, it's actually been around for a while. At least in the United States, the kind of initial recognition of issues with 6-PPD-quinone is associated with coho salmon mass die-offs in the Pacific Northwest, especially around Puget Sound. Since 2000, the exact chemical cause at that point, since 2000, when it was first, this urban runoff mortality syndrome was observed until the very recent, in 2020, the exact chemical cause was unknown, but the impact was recognized already. And so researchers, it took them a while to figure out what exactly was causing this urban runoff mortality syndrome. And then in 2020, 6-PPD-Q was identified as a causative agent. And I've included this citation here to Tian et al. 2020. This is the seminal paper that makes the link between 6-PPD-quinone and impacts on coho and other salmon. So why is there concern about 6-PPD-quinone? Just, I kind of alluded to it already, but it's toxic at very low concentrations to several salmon species, in particular to coho salmon, and the lethal concentrations to coho salmon are in the range of 0.04 to 0.095 micrograms per liter, which is in parts per trillion. So this is a very toxic chemical. Additionally, 6-PPD-quinone is occurring in the environment at relevant concentrations. So frequently in Puget Sound and streams running into or flowing into Puget Sound, we see concentrations of 6-PPD-quinone around or above 0.09 micrograms per liter. So this is coho are being exposed to lethal concentrations routinely. And so it's a major issue, at least as observed in Puget Sound. Additionally, 6-PPD has been tested broadly and observed broadly in the US in surface waters and in airborne particulate matter. And it's still widely released. Tires are still produced and used in the United States containing 6-PPD and 6-PPD-quinone. And this is, in terms of an issue in EPA Region 9, our geographical area. So coho salmon, the southern extent of coho salmon goes down to the central coast of California. So this is potentially an issue in the central coast of California and especially in the north coast of California. I'll just quickly go over. This is the conceptual transport and exposure model for 6-PPD-quinone. Large storm events convey 6-PPD-quinone from roadways to aquatic systems. Additionally, 6-PPD-quinone is made airborne for potential inhalation and deposited on soil and plants for potential uptake through ingestion for people who are close to the soil and close to plants. In addition to routine tire wear, 6-PPD-quinone enters the environment through scrap tires, recycled tire crumb materials, and rubber modified asphalt. We don't know much about the bioaccumulation of 6-PPD at this point. We don't know that it does bioaccumulate, but there just has not been very much research into that yet. Okay, I've talked about this briefly. I'm going to go into a little bit more detail. So the research that's been done so far has focused on, the toxicity research that has been done so far is focused on salmon. There have been a few other species as well, several fish, and a couple of aquatic invertebrates. But for the most part, the toxicity studies have focused on salmon. And what we see is there's several sensitive species, salmon species, including coho, char, brook, and rainbow trout. The species that have been determined as not sensitive to 6-PPD-quinone are sockeye, Arctic char, Atlantic salmon, and brown trout. And then I reserved kind of the separate note for Chinook salmon. Chinook has not been identified as sensitive in lab tests, but it has demonstrated mortality when exposed to roadway runoff. And it's not known exactly what the chemical cocktail was in that roadway runoff, but Chinook were observed to be sensitive to roadway runoff concentrations. Yes, so mammals, birds, and amphibians mostly are untested. So we don't know that they are sensitive, but we don't know that they're not sensitive either. So that's an area of additional research that's being pursued. And then I just also wanted to mention the loss of coho not only impacts coho, but it impacts food webs and inland nutrient transfer as coho would have made their way inland for spawning and dying there and releasing their fertilizer upstream.
How come do you think it's not affecting the reds or the kings as much as it is the silvers?
The mode of action appears to be 6-PPD-quinone getting through the blood-brain barrier. And so for whatever reason, it's a lot easier for 6-PPD to do that in coho salmon. And I don't know the physiological reasons for that.
Yeah, it seems strange. If it didn't want it, you'd have them all, you think. I mean, there's a lot less reds around, more kings with the silvers.
Yeah, no, it's a good point, and I think that's a question that the researchers are asking. And in terms of I think the toxicity tests on salmon, there's going to be more of those tests to try to. I don't think we did pinks. I don't see chums up here either. So right, there's the toxicity testing that's been done so far is on a limited number of species. Yes. And I just brought this table up, and I'm crediting the Interstate Technology Regulatory Council fact sheet for this. But you can see the relative toxicity of these different species. And you can see coho is, approximately an order of magnitude more sensitive than char and brook trout and rainbow trout. But those, those categories 2 through 4, those rows 2 through 4 are still sensitive because they're not that far. Those toxicity values aren't that far above what you see 6-PPD concentrations that you see in the environment. And I'm just going to mention, because I didn't include a website for the Interstate Technology Regulatory Council. They have an excellent website on 6-PPD-quinone. They've got a recently released fact sheet, and apparently they're going to be releasing additional support documents. Yes. I don't know that we've tested those. I don't think we have, but I don't know for sure. Right. Yes, thank you. Okay, so these are sublethal effects, and so basically there has not been a lot of research on sublethal or chronic effects. A couple of studies on zebrafish and roundworm and mice shows that there is potentially sublethal effects at relatively low concentrations. Okay, moving on to human health effects. So again, not a lot of information yet, but all of these questions are in the offing for research that's going to be starting up or is already ongoing. Toxicity to humans is mostly unknown at this point. There is likely increased risk of 6-PPD-quinone to high consumers of fish, although that's not yet characterized. There are a number of studies on the bioaccumulative properties of 6-PPD, but those studies are still limited, and that research results haven't been determined yet. One thing that's kind of interesting is that there was a study in China that identified the urine of pregnant women having the highest levels of 6-PPD-quinone. So there's some indication that pregnant women are storing 6-PPD-quinone to some extent. Okay, so additionally, EPA and other agencies are studying tire wear particles in general. and this is important, just to kind of a couple of factoids here. One tire shreds between 2 to 14 pounds of particles in its base road wear. And these tire wear particles, tire wear composition includes microplastics, heavy metals, hydrocarbons, and other toxic chemicals, which can then be transported to soil, water, and air. Research is underway to characterize the effects on aquatic and human health of tire wear particles. So not just looking at 6-PPD and 6-PPD-quinone, but looking at some of these other chemical makeup materials of tires. Comprehensive cleanup and risk reduction solutions are needed as well. So, and I just want to point to this one article that's posted on EPA's website by Paul Mayer et al. And this is a really good article that just kind of sums up the existing research on tire wear particle effects. And there's also, what's nice is at that EPA website, there's also a recording where Paul goes through a slide presentation on that article. I won't spend much time on this slide, but it kind of summarizes what I just mentioned, which is that, you know, you've got this cycle of tire wear production, release into the environment. What do you do with these tires once they have used up their utility as for cars? They have to go somewhere. If you recycle them, it's always good to recycle, but there are concerns about recycled tire byproducts getting into the environment as well. Okay, impacts on tribes. And I just wanted to mention these really quickly. They include the economic impacts of declining salmon populations on tribal commercial fishers, loss of coho salmon and other salmon species as a dietary staple due to 6-PPD-quinone, potential heightened ingestion risk due to bioaccumulation. Again, that's still not known, but that's something that EPA and other researchers or other agencies are looking into. Waterborne exposure as part of cultural, recreational, and subsistence activities. So just being out on the water, potentially there could be waterborne effects for those people who are out on the water a lot. And additionally, exposure to soil and plant-laden 6-PPD in food gathering and medicine practices could be a potential route for 6-PPD exposure. Okay, so we've got this, I'm really excited about this particular project, partly because I'm engaged in it, but this is a project as part of the Office of Research and Development, EPA's ROXNet program, which is an opportunity for tribes, states, and EPA regions to connect with EPA's Office of Research and Development to work with scientists there on research needs. So we've pulled together this team to look at 6-PPD-quinone impacts on California North Coast waters, especially impacting coho salmon. And membership on that team includes folks from Yurok, Pinolville, Pomo, and Wiyot tribes, along with California State Board folks and EPA staff, including myself and Matt. So we're focusing in on the North Coast area, and we also are connected with a broader team of tribal folks from all of the North Coast portion of California who are helping to inform the ROXNet team of their concerns and research needs that they see on 6-PPD-quinone. Okay. Okay, just super quick on this slide, and you can, I won't say too much here, but these are recent EPA activities on 6PPD and tire wear particles. In May 2024, EPA published screening values. These are like aquatic life criteria values for 6PPD and 6PPD-quinone. In November 2023, EPA granted a petition submitted by multiple West Coast tribes to consider establishing regulations prohibiting the manufacturing of 6PPD in tires, and EPA intends to publish an advance notice of proposed rulemaking in the fall of 2024 to gather more information to inform a possible regulatory action. In March 2024, EPA made a big award to Puget Sound stormwater funding, and in December 2023, EPA published an analytical procedure to test for 6-PPD-quinone. I think this is the second to last slide, and I'll just leave it here so I think you all are going to hopefully have access to these slides. Okay, so I'll just leave this slide here. You can go to those websites. And mostly I want to thank you very much for listening to this presentation and giving me the opportunity to share it with you.
Can we do one or two questions, Anna, or are we done?
Jeanette, we are out of time.
Okay, very quickly. I think you had your hand up first, and then we'll go to Dominic.
I'm Leslie Cleveland, and I'm with
Come
the
on up,
Bureau of
Alice.
Reclamation. I have just two quick, broad questions. One is, are you the only lab testing for the aquatic, specifically the salmon, or because we have a big salmon in Alaska in halibut. So is there a different lab and you guys talk to each other, or is your lab looking at the whole West Coast?
We're not currently analyzing for 6-PPD.
Okay.
Region 10's lab is analyzing. We're trying to figure out right now if that makes sense for our lab. Just real quick, Alex.
And do you talk to each other? And then just real
Yes.
quick, because it's a follow-on to, is that what about we export very, high amounts of salmon, export it, but we also get imports. So are you testing the imported salmon coming into specifically our area?
So I think the answer is I don't know of any testing like that's going on. And I'm assuming this is in terms of, farm-raised salmon from some other part of the planet. Yeah.
Who is it? Oh,
Yeah, I was just wondering if the 6-PPD is persistent or if it has some half-life where it breaks down.
I think initial studies is that it's not super persistent. I think the concern is it's continually being reintroduced into the environment through new tires and new tire wear. So it's ongoing. And so in that regard, it's persistent.
And I want to just add that there's in the category of no free lunch, the electric vehicles burn through tires 2 to 4 times faster because they are much heavier as a result of the batteries. Folks, I really want to thank everybody for coming today. Oh, one more.
One more quick question.
Okay, Paul.
Is there any research being done on the exact mechanism of the toxicity or the mortality?
There is, and I alluded to it earlier. They think that 6-PPD is getting into, it crosses the blood-brain barrier and it gets into the brain and it causes behavioral, really adverse behavioral reaction. And that's an ongoing area of research. So I don't think they know the answer to that quite yet. Yeah.
Thanks again, folks, for coming. Please come to the next session where we're going to talk about how you can get funding for treatment and for testing for PFAS and for emerging contaminants.